Good morning. My name is Yuchang, and I'm a postdoc in Chung's group at the University of Wisconsin-Madison. It's my great pleasure to share with you our project that uses GWAS on birth year infant mortality rates to interrogate the natural selection traces hidden in the human genome. This is a collaboration work with Shiro, Zihang, Jenna, and supervised jointly by Jason and Chungshi. Quantifying recent natural selection in human populations is vital for the understanding of the history of our own species. Currently, there are two types of studies that can identify the selection signal at the SNP level. The first one is represented by Madison et al.'s Nature paper that compare allele frequencies across populations. The second type is developed by Field et al. Science paper that can infer selection signals through identifying unusually long haplotypes as the result of rapid changes in allele frequencies. But both studies suffer from limited sample sizes and large uncertainties in pinpointing the timing of selection. In most cases, we could only know that the selection happened over the time span of several thousand years. In this project, we advanced the field by showing that GWAS can be used to identify recent selection signals. We demonstrate evidence for selection at the two loci in the past century. We deployed a novel regional GWAS to quantify the association between each SNP and the infant mortality rate IMR. We have each year's IMR record for each county in the UK. We assign each UK biobank sample an IMR value by linking its birth year and birthplace with regional IMR record. IMR is a well-established indicator of area-level conditions related to nutrition, infection, and inflammation. It therefore indexes the environment experienced during gestation and infancy. We ask which SNP alleles are sustained in particularly harsh environments and test how surviving SNPs are correlated with an array of complex traits. Here, panel A shows the average IMR value between 1936 and 1970 for each county in the UK, England and Wales. Panel B shows the IMR record for each county and each year. Panel C and D are the distributions of IMR among GWAS samples. These are the Manhattan and QQ plots for the IMR GWAS. We got two genome-wide significant hits, the LCT locus on chromosome 2 and the TLR16 gene cluster on chromosome 4. Both loci have very convincing LD support as shown in the locus zoom plots. These two loci were also identified as selection targets in previous publications. In the bar plots shown in the middle, we show the association with birth year IMR at 12 target loci for selection in Europeans. Genetic loci for pigmentation and eye color showed substantially weaker association with IMR compared to loci associated with lactase persistence, immunity, and vitamin D metabolism. We also run IMR GWAS for each year. The figure on the right shows the effect sizes of two of these SNPs. The effect sizes peaked around 1942 and 3 and eventually bounced back to 0, suggesting larger selection pressure during the World War II. We next examine the genetic correlations between birth year IMR and a set of 50 traits widely assessed as outcomes of selection processes. Multiple significant genetic correlations were found. Among known targets of selection, we found significant correlations with vitamin D, but no result with hair color or skin color. Our results are consistent with other approaches showing correlations with genetics for fertility, age at first birth, but not with number of children, sporadic miscarriage, age at menarche, and age at menopause. Similar to earlier findings, we show correlations with EA and cognitive performance, but extend this finding by showing these results are driven by the direct components, not the indirect component. To conclude, we replicated the two loci of natural selection targets using a novel regional GWAS on infant mortality rates. Selection pressure peaked at 1942 and 3 during World War II, suggesting the ongoing selection in a modern population. Significant genetic correlations between IMR and a host of phenotypic domains reinforce earlier findings related to fertility, anthropometrics, cognition, and also extending findings into psychiatric and health conditions. Finally, we showcase that GWAS can be used as a powerful alternative alternative approach to identify natural selections on the SNP level. Thank you very much for your attention.